Stop. Before you reach for another bottle of biotin, before you book another consultation about minoxidil or finasteride, before you spend another dollar on a shampoo that promises thickness and delivers nothing, I need you to watch this video because what I am about to share with you is the piece of the hair loss puzzle that virtually every dermatologist, every trichologist, and every supplement company is either unaware of or deliberately not talking about. And once you understand it, everything you thought you knew about why you are losing hair is going to shift completely. This is not about DHT. It is not about genetics. It is not about stress, at least not in the way you have been told. It is about something happening silently inside your body right now, something measurable, something reversible, and something that, if left unaddressed, will continue to accelerate your hair loss no matter how many supplements you take or how expensive your shampoo is. If you are new to this channel, I am a physician and researcher who has spent decades studying how the aging body, specifically the body over 50, responds differently to nutritional deficiencies, hormonal shifts, and inflammatory processes than younger bodies do. The science I am about to share is real, it is referenced, and the studies are linked in the description below. Before we dive in, I want to hear from you personally. Tell me in the comments right now, how long have you been experiencing hair loss or thinning? Are you a man or a woman? And what have you already tried? Drop your age and your answer below. I read every single comment. Everyone. And your experiences directly shape the content I create for this community. Here is what we are covering today. I am going to walk you through exactly what the number one missing factor in hair loss is, the biological mechanism behind why it causes hair to fall, why adults over 50 are catastrophically more vulnerable to this than younger people, and the precise evidence-based steps you can take this week to begin reversing the damage. We are counting down from 5 to 1. 5 reasons this missing factor is destroying your hair. We say to satiric in tricancer, ranked from important all the way up to the single most critical piece of information that could change your hair and your health simultaneously. And I will tell you right now, number 1 is something almost no one in the hair loss space is addressing. The reason why is going to make you very frustrated and then very motivated. Let us begin. The number one missing factor in hair loss is chronic scalp and systemic microinflammation, not the inflammation you feel when you injure yourself, not the kind that swells and hurts and visibly announces itself, the invisible kind, the silent, low-grade, persistent inflammatory state that burns through your body at the cellular level for years, sometimes decades, before its effects become impossible to ignore. And by the time you are watching hair fall into the shower drain every morning, that fire has already been burning for a long time. Think of your hair follicle this way. Imagine a flower growing in soil. The flower is your hair strand, the soil is your scalp tissue, and the microvascular network, the tiny blood vessels that feed each follicle. When that soil is rich, oxygenated, anti-inflammatory, and nutrient-dense, the flower grows. Strong, thick, rooted deeply. But when that soil becomes toxic, when inflammatory compounds saturate the tissue around the follicle, when blood flow is restricted, when the cellular environment turns hostile, the flower shrinks. The root weakens, eventually it stops producing entirely. And no amount of fertilizer dropped on top of a poisoned root system will bring that flower back. That is what is happening in the scalps of millions of adults right now. And almost nobody is treating the soil. Let us count down. Number 5, microinflammation destroys the dermal papilla, the command center of your hair follicle, and mainstream hair loss treatments do not touch it. The dermal papilla is a cluster of specialized cells sitting at the very base of each hair follicle. It is the biological headquarters of hair growth. It receives signals from your bloodstream, communicates with the follicle stem cells above it, and controls whether a follicle is in the growth phase, called anagen, or the dormant and shedding phases, called catagen and telogen. When the dermal papilla is healthy, hair cycles normally. When it is under sustained inflammatory attack, it begins to malfunction. Growth cycles shorten, dormancy periods lengthen, and over time, the follicle miniaturizes, producing progressively thinner, shorter, weaker strands until it stops producing hair entirely. A landmark study published in the Journal of Investigative Dermatology identified elevated levels of prostaglandin D2, a powerful pro-inflammatory signaling molecule, in the scalp tissue of men and women with androgenetic alopecia, the most common form of hair loss in adults over 50. The concentration of this inflammatory compound was significantly higher around miniaturizing follicles than around healthy ones. And critically, this inflammatory environment was present independently of DHT levels, meaning it was not simply a downstream effect of the hormone most commonly blamed for hair loss. It was its own parallel destructive process. After the age of 55, your body's production of anti-inflammatory compounds, specifically lipoxins and resolvins, which are responsible for resolving and switching off inflammatory responses, declines significantly, according to research from Harvard Medical School's Department of Immunology. Your inflammatory response becomes, in a very real sense, harder to turn off. The fire burns longer. The dermal papilla suffers more sustained damage per inflammatory episode, and the cumulative effect on the follicle over years is profound. The synergy tip here: omega-3 fatty acids, specifically EPA and DHA from fatty fish, are direct precursors to the resolvins and lipoxins your body needs to resolve scalp inflammation. 3 servings of wild salmon or sardines per week provides clinically relevant amounts. This is not background nutritional advice. For an adult over 55 with hair thinning, this is a targeted anti-inflammatory intervention aimed directly at the dermal papilla environment. Number 4. The gut-scalp axis, the connection almost nobody in dermatology is talking about. I want to introduce you to a relationship that is reshaping how the most advanced researchers in the world think about skin and hair disorders. It is called the gut-skin axis, and for hair loss specifically, the gut-scalp connection is emerging as one of the most significant and most overlooked pathways in the entire field. Here is the mechanism. Your gut microbiome, the trillions of bacteria living in your digestive tract, is responsible for regulating systemic inflammation throughout your body. A diverse, healthy microbiome produces short-chain fatty acids, particularly butyrate, that directly suppress inflammatory signaling and maintain the integrity of your gut lining. When the microbiome is depleted, and when beneficial bacteria populations collapse and pathogenic strains dominate, a state called dysbiosis, the gut lining becomes permeable. Inflammatory compounds leak into the bloodstream, and that systemic inflammatory burden lands everywhere, including your scalp. After the age of 60, gut microbiome diversity declines measurably in the majority of adults. A study published in the journal Cell Host and Microbe found that older adults with the lowest microbiome diversity had systemic inflammatory marker levels, specifically interleukin-6 and C-reactive protein, that were 2 to 3 times higher than age-matched peers with diverse microbiomes. These are the same inflammatory markers that the dermal papilla research identifies as drivers of follicle miniaturization. A 2021 study published in Experimental Dermatology found that patients with alopecia areata, an autoimmune form of hair loss, had significantly altered gut microbiome profiles compared to controls with dramatically reduced populations of lactobacillus and bifidobacterium and elevated populations of inflammatory bacterial strains. When researchers in a separate trial administered probiotic supplementation to restore microbiome diversity in alopecia patients, they documented measurable improvements in hair density and reductions in scalp inflammatory markers over a 6-month period. I think about a patient I will call Diane. She was 63 years old, a retired teacher from Columbus, Ohio. She had been experiencing progressive hair thinning for 4 years. She had tried biotin at high doses. She had tried a DHT blocking shampoo. She had tried collagen peptides. Nothing had made a meaningful difference. When I looked at her full picture, not just her hair, I found what I find so often in this demographic: significant gut dysbiosis, elevated CRP, a diet high in processed carbohydrates and low in fermented foods. We restructured her nutrition around gut restoration as the primary goal. Within 5 months, her dermatologist noted visible improvement in hair density in the crown region. Her inflammatory markers had dropped substantially. She told me, "I came to you about my hair and you fixed my whole body." She was not wrong, because the gut and the scalp are not separate systems. They are downstream from the same river. The synergy tip: add 1 serving of fermented food daily: plain kefir, kimchi, sauerkraut, or plain yogurt with live active cultures. Simultaneously, eliminate or dramatically reduce refined sugar, which research from the Weizmann Institute shows actively depletes beneficial gut bacteria within 48 hours of consumption. These 2 changes together produce a microbiome shift that begins within 72 hours and measurably reduces systemic inflammatory burden within 4 to 6 weeks. Number 3. Chronic scalp hypoxia, the blood flow crisis silently suffocating your follicles. Your hair follicles are among the most metabolically active structures in the human body. During the anagen growth phase, a single follicle consumes more oxygen and nutrients per unit of tissue than almost any other structure outside of the brain. It needs an extraordinary supply of blood flow to sustain active hair production. And for adults over 60, that supply is being quietly choked off. After the age of 60, scalp microvascular density, the number and functional capacity of tiny blood vessels supplying the scalp, declines by an estimated 40% compared to a 30-year-old, according to research published in the British Journal of Dermatology. This reduction is driven by a combination of chronic low-grade inflammation, damaging the endothelial cells lining blood vessels, declining production of nitric oxide, your body's natural vasodilator, and reduced angiogenic signaling, meaning your body becomes less efficient at maintaining and regenerating the microvascular networks that feed follicles. When a follicle is hypoxic, starved of oxygen and nutrients, it enters a stress response. Growth cycles shorten. The follicle begins prioritizing survival over production, and the hair it does produce becomes thinner, weaker, and more prone to breakage and shedding. A right pause here. If this information is reaching you and it is clarifying something you have felt but could not explain, why your hair feels weaker, why volume is disappearing even though you are doing everything right, please hit the subscribe button and the like button right now. I publish new evidence-based content every week specifically for adults over 60. The YouTube algorithm only amplifies these videos when you engage with them. Please like, subscribe, and share this with someone you know who is struggling with hair loss. It costs nothing, and it could genuinely change what happens next for them. Back to number 3. The most evidence-supported dietary intervention for scalp microvascular restoration is not a supplement. It is dietary nitrate, found in abundance in beetroot, leafy greens including spinach and arugula, and dark leafy vegetables like Swiss chard. When you consume dietary nitrates, gut bacteria convert them to nitrite and then to nitric oxide, the compound that relaxes and dilates blood vessel walls, improving blood flow to peripheral tissues, including the scalp. A study published in the Journal of Applied Physiology found that dietary nitrate supplementation significantly improved microvascular blood flow to skin and scalp tissue in adults over 60 within 2 weeks of consistent consumption. Combine this with daily scalp massage, 4 to 5 minutes of firm circular pressure using fingertips, ideally with a few drops of rosemary oil diluted in a carrier, and you create a compounding effect. A randomized controlled trial published in Skin Pharmacology and Physiology found that participants who performed consistent scalp massage over 24 weeks showed measurable increases in hair thickness attributable to improved blood flow and mechanical stimulation of dermal papilla cells. Rosemary oil specifically has been studied in a head-to-head clinical trial against minoxidil, the most widely used pharmaceutical hair loss treatment, and produced equivalent hair count improvements at 6 months with 0 of the scalp itching and irritation side effects that minoxidil commonly causes. The synergy tip: consume a beetroot-based smoothie or a large spinach and arugula salad daily for dietary nitrate, and perform your scalp massage in the morning within 30 minutes of waking when cortisol and circulation are naturally peaking. Morning cortisol elevates vascular tone transiently. Pairing mechanical stimulation with this natural circulatory peak amplifies blood flow to follicles during the window when they are most receptive. Number 2, the vitamin D and hair follicle receptor crisis that is accelerating hair loss in 70% of adults over 60. I need you to hear this number. According to data from the National Health and Nutrition Examination Survey, approximately 70% of adults over the age of 60 in the United States are either deficient or insufficient in vitamin D, 70%. And the implications for hair loss are direct, measurable, and almost entirely ignored in mainstream conversations about thinning hair. Here's the biology. Vitamin D does not just regulate calcium absorption and bone health. Vitamin D is a steroid hormone that binds to vitamin D receptors, called VDRs, present in virtually every tissue in the body, including, critically, the hair follicle, specifically in the outer root sheath keratinocytes and the dermal papilla cells. When vitamin D binds to these receptors, it promotes the transition of follicles from the dormant telogen phase back into the active anagen growth phase. It also regulates the expression of genes involved in follicle cycling, keratinocyte differentiation, and, most importantly, the suppression of local inflammatory signaling around the follicle. When VDR signaling is impaired, either because vitamin D levels are inadequate or because VDR expression itself has declined, which occurs with aging, follicle cycling becomes dysregulated. Dormancy periods lengthen. The follicle spends more time not growing than growing. And over months and years, this imbalance compounds into visible progressive thinning. A meta-analysis published in the journal Dermatology and Therapy reviewed 11 studies examining the relationship between vitamin D levels and hair loss disorders. The pooled analysis found a significant association between low serum vitamin D and alopecia areata, telogen effluvium, and female pattern hair loss across all age groups, with the association strongest in adults over 50. A 2019 randomized controlled trial published in Skin Appendage Disorders found that vitamin D supplementation in deficient patients with telogen effluvium, stress-related diffuse hair shedding, produced statistically significant reductions in daily hair loss compared to placebo over 3 months. After the age of 65, your skin's capacity to synthesize vitamin D from sunlight declines by approximately 75% compared to a 20-year-old, according to research from the University of Boston School of Medicine. The same sun exposure that was once adequate for your vitamin D needs is now producing a fraction of what your body requires. And dietary sources alone, fatty fish, egg yolks, fortified foods, are inadequate to compensate for that deficit in most adults. The practical implication? If you are experiencing hair loss and you have not had your serum 25-hydroxyvitamin D level checked recently, that blood test should be your first phone call tomorrow morning. The optimal range for follicle health based on the research literature is between 50 and 80 nanograms per milliliter, significantly above the often cited clinical minimum of 30. Most adults over 60 who have not been supplementing are well below 40. Supplementation dosing should be individualized with your physician based on your baseline level, but the research generally supports 2,000 to 4,000 IU daily for maintenance in adults over 60, with higher therapeutic doses for deficiency correction under medical supervision. The synergy tip: take your vitamin D supplement with your largest meal of the day alongside a source of healthy fat, olive oil, avocado, salmon, because vitamin D is fat soluble and absorption increases dramatically when consumed with dietary fat. Additionally, combine vitamin D with vitamin K2 supplementation, 100 to 200 micrograms of MK7 form daily, because K2 directs the calcium that vitamin D mobilizes to your bones rather than your blood vessels, protecting vascular health and maintaining the microvascular integrity your scalp depends on. And now, number 1, the single most important missing factor in hair loss, and why almost no one is addressing it. Are you ready? Because this is the piece that ties everything together. And it is the reason why people who address DHT alone, who take biotin alone, who use minoxidil alone, so often see partial results at best or no results at all. The number 1 missing factor is the disruption of the hypothalamic pituitary adrenal axis, your body's central stress response system, and its direct documented catastrophic effect on the hair follicle cycle. I know. You have been told stress causes hair loss, but what you have not been told is the precise biological mechanism. How chronic HPA axis dysregulation is, which is not the same as feeling stressed, literally chemically signals your hair follicles to stop growing. And why adults over 60 are uniquely and severely vulnerable to this process in a way that younger people simply are not. Here is the mechanism. When your HPA axis is chronically activated by sustained psychological stress, by poor sleep, by systemic inflammation, by nutritional deficiency, by social isolation, by any chronic physiological threat, it produces sustained elevated levels of cortisol and corticotropin-releasing hormone, or CRH. CRH receptors are expressed directly on hair follicle cells. And when CRH binds to those receptors, the follicle receives a direct molecular signal to enter catagen, the regression phase, and cease active hair production. This is not a metaphor. This is a direct hormonal signal telling your follicle to stop growing. A landmark study published in the American Journal of Pathology introduced CRH directly into mouse hair follicles and documented immediate premature entry into catagen and follicle miniaturization within days. In human studies, patients with chronic HPA axis dysregulation, documented through cortisol measurements, showed disproportionately high rates of telogen effluvium and accelerated androgenetic alopecia progression compared to age-matched peers with normal cortisol rhythms. Now, here is why this is uniquely devastating for adults over 60. After the age of 60, the HPA axis loses something called negative feedback sensitivity. In a young, healthy body, cortisol levels rise in response to a stressor and then fall as the HPA axis self-regulates. In an aging body, this self-regulation becomes impaired. Cortisol stays elevated longer after each stressor. The baseline cortisol floor rises, and the compounding effect of decades of stress events, poor sleep, inflammatory burden, and loneliness, which, as research from Brigham Young University confirms, is itself a biological stressor affecting nearly 43% of adults over 60, creates a sustained HPA axis activation that never fully resolves. Your follicles are living in a body that is chronically telling them to stop growing, and no topical treatment, no supplement, and no shampoo addresses that signal. I want to tell you about Martin. He was 71 years old, retired architect from Seattle, Washington. He had been losing hair steadily for 6 years. He had tried every evidence-based topical treatment available. He had optimized his nutrition. He exercised. His DHT levels were well managed, and yet his hair continued to thin. When we did a full hormonal workup, his diurnal cortisol curve was severely blunted. His morning cortisol peak was inadequate, and his evening cortisol was elevated, the signature pattern of chronic HPA axis dysregulation. His sleep architecture was disrupted. His social connections had narrowed significantly since retirement, and his inflammatory markers told a story of a body under sustained systemic stress. We addressed each element systematically. Sleep optimization using the circadian reset protocol. Zone 2, cardiovascular exercise 4 times weekly to restore cortisol regulation and produce brain-derived neurotrophic factor that supports HPA axis resilience. Structured social engagement. He joined a weekly architecture lecture series and a Saturday morning walking group. Daily adaptogenic support through ashwagandha root extract, which a 2019 double-blind trial published in Medicine showed significantly reduced serum cortisol and improved stress resilience scores over 60 days. And targeted nutritional anti-inflammatory support. Within 4 months, Martin's cortisol rhythm had normalized measurably. Within 6 months, his hair loss had slowed dramatically. At 8 months, his barber commented unprompted that he was seeing new growth in areas that had been thinning for years. He told me something I will not forget. He said, "Nobody ever asked me how I was sleeping or how lonely I had been. Everyone just looked at my head. That is the problem. Everyone is looking at the head. Nobody is looking at the body the head is attached to." Here's your action plan starting this week. Get your vitamin D and cortisol levels tested. Ask your physician for a serum 25-hydroxyvitamin D and a diurnal salivary cortisol panel. Address gut inflammation through daily fermented foods and elimination of refined sugar. Consume fatty fish 3 times weekly and a nitrate-rich leafy green salad daily. Perform 4 minutes of scalp massage every morning. Optimize your sleep using a strict circadian protocol. Begin Zone 2, exercise 3 to 4 times weekly. And treat your social connections as a medical intervention, because that is exactly what the science says they are. Your hair is not failing you. Your body has been sending a distress signal for years, and nobody handed you the translation. Now you have it. If this video gave you clarity that you have not found anywhere else, and I hope it did, please subscribe to this channel right now. Every single week, I bring you information like this that is specifically researched and specifically designed for adults over 60. Share this video with one person today, someone who has been struggling, someone who deserves to understand what is actually happening. And tell me in the comments, which of these 5 factors do you think is most responsible for your hair loss? And are you going to get your vitamin D tested this week? I will be reading every single response. Thank you for being here.